The human body is made up of thousands of cells containing structures that are necessary to perform everyday functions in our lives, including our DNA, which stands for deoxyribonucleic acid. The DNA inside our bodies is what makes us tick. It holds our hereditary information and makes us who we are.
DNA is shaped like a ladder that's been twisted, which is called a double helix. The steps of the ladder are made of four main chemicals called bases: adenine, thymine, guanine, and cytosine. The particular order of A's, T's, G's, and C's is extremely important.
The combination of these units of adenine, thymine, guanine, and cytosine as they pair with each other varies from person to person. It makes us unique as it pulls together our genome. DNA is made up of discrete units of hereditary information consisting of specific nucleotide sequences called genes. In other words, genes are the unique sequences of DNA's 4 nucleotides. Genes help to make up this genetic code in our bodies, which helps to regulate and carry out processes. DNA, the genes it's composed of, and its associated proteins do not just float around freely in the cell. If this happened, then nothing would get done. Instead, it is contained in the nucleus in structures called chromosomes. DNA and its genes are condensed down into chromosomes. Somatic cells are diploid and contain 46 of these chromosomes. On the other hand, sperm and egg cells are haploid and contain 23 of these chromosomes. A permanent change in the unique sequences of nucleotide base pairs in these chromosomes can cause dramatic changes to the processes inside the human body. There are 3 different types of mutations: point mutations, deletions, and insertions. For example, our DNA is like a recipe. As you can see, a point mutation has occurred where 2 eggs is changed to 2 egger, or EGGR, instead of EGGS. In this type of mutation, a single base nucleotide in the DNA is replaced with another nucleotide of the genetic material. As you can see, this changes the recipe, but the cook can still see what to do, and he's probably going to make some pretty good chocolate chip cookies. For insertions. An insertion has occurred where it says 21 eggs instead of 2 eggs. In this type of mutation, there is an addition of extra DNA into a section of the genetic material. Imagine adding 21 eggs to a batch of cookies. Talk about gross. This dramatically changes the recipe. Deletions. In this example, we see deletion where step 1 of the instructions is missing. Deletion is when a sequence of DNA is missing, just the opposite of insertion. Making cookies without preheating the oven won't do much. All you will come out with is raw cookie dough. A deletion can cause dramatic changes to our internal recipes. These dramatic changes or mutations to the DNA sequences in a gene can occur in 2 different ways. A person may inherit this mutation from their parents, or the mutation may be acquired through a person's life. When the mutation is inherited from one's parents, it is called a hereditary or germline mutation, because they are present in both the egg and sperm cells. In recent discoveries, it has been found that the molecular basis for a type of cancer, paraganglioma, is a germline mutation. Paraganglioma can form in many different places of the human body, including the neck and the abdomen if the patient has this specific mutated gene. These germline mutations can occur on up to 6 different genes, including the gene encoding for succinate dehydrogenase, or SDH, with subunits of B, C, and D. These genes map specifically to chromosome 11, which we will talk about in more detail shortly. Mutations in this specific gene account for about 70% of hereditary paragangliomas. SDHD and SDHB mutations most often lead to head and neck paragangliomas, including the carotid body, which is known as the most common site for paragangliomas. In this type of paraganglioma, the tumor is located between the internal carotid artery and the external carotid artery, which become encased by the tumor as it enlarges. Just to give you an idea of where everything is, the external carotid artery is on the top, and the internal carotid artery is on the bottom. And this is also just a picture of how big the tumor was. On the SDHD gene, the majority of mutations are point mutations and deletions. Although they are very rare, other types of alterations other than point mutations and deletions have been found. 50% of all paraganglioma cases are hereditary, and also show autosomal dominant inheritance of the SDHD gene. Autosomal refers to a non-sex chromosome. Autosomal dominance is when one particular form of a gene, one allele, dominates over other alleles and is always expressed when present in an individual, whether they are homozygous for that allele or heterozygous. Paraganglioma is an odd case because it has an incomplete penetrance on the SDHD gene mutation, depending on whether the gene was inherited from the mother or from the father, even though it is always inherited dominantly. An offspring is only susceptible to forming paraganglioma if the gene is inherited from the father. In this case, they may also pass the gene to their offspring as well. If the gene is inherited from the mother, then the offspring do not produce paraganglioma, but instead, they only have the capacity to pass this gene along to their offspring. This type of effect occurs because of genetic imprinting. The SDHD gene is only activated during spermatogenesis, which is the process of producing sperm with half the number of chromosomes as somatic cells. Paraganglioma is a tricky disease to grasp. In the next few minutes, we will just focus on the SDHD gene mutation. As covered before, human cells have 46 chromosomes, or 23 pairs. Of those 23 pairs, the SDHD gene is located on chromosome 11 at position 23. What that means is that the SDHD gene location is from base pair 111,957,570 to base pair 111,966,517 on the 11th chromosome. Before we go into how the SDHD gene mutation actually creates the paraganglioma cancer, we need to understand what the SDHD gene normally does. The SDHD gene is one of the 4 subunits responsible for producing succinate dehydrogenase, or SDH, in the mitochondria. The SDHD protein helps hold the SDH enzyme in place during the citric acid cycle and oxidative phosphorylation, 2 key pathways for energy conversion. In these 2 processes, the SDHD gene acts as a middleman. First, in the citric acid cycle, SDH helps to convert succinate to fumarate. During this reaction, electrons are released and the SDHD protein transfers them to the next step, which is oxidative phosphorylation. During oxidative phosphorylation, the electrons are used in order to help create energy in the form of ATP, which is a main source of energy for our cells. Most inherited SDH gene mutations change amino acids in the SDH protein sequence or result in a shortened protein. This means that there is little to no SDH enzyme activity. Because the SDH enzyme cannot convert succinate to fumarate, succinate accumulates in the cell. The excess succinate abnormally stabilizes HIFs, which are hypoxia-inducible factors. HIFs are transcription factors that respond to changes in available oxygen in the cellular environment. In response, HIF also builds up in the cell. Excess HIF triggers the cells to divide, which causes the production of blood vessels when they are not needed. This rapid and uncontrolled cell division can lead to the development of tumors in people with paraganglioma. All in all, we can see that the production of paraganglioma results in the smallest of mutations on a gene. This shows that sometimes the smallest things have the biggest impact.